All right. Well, thank you again, Alexander, for joining us this week. It's a real pleasure and honor to have someone who knows so much about the coronavirus joining us. And I thought one of the initial questions that I might ask is maybe just kind of about history of taxonomy. The paper that you've shared with us talks about the recent history of taxonomy of viruses and how there was a switch from the use of I guess, cross-reactivity of the spike protein to more recent genomic techniques. And you also had a really interesting discussion in there about just this broader shift from initially viral taxonomy was driven by pathology and driven by a disease. And now you see a real shift towards phenotypic free classification. So when was the first coronavirus described? How does this fit within broader viral, histories of viral taxonomy?
Yeah. Thanks again, Eben, for inviting me to discuss. And I really amazed on the range of the expertise in this group and interest. And also, I really appreciate that you're interested in this particular paper from the angle of species, which is not very common, unfortunately, in virology. If you look in the publications, you barely find mentioning of species. So what we wrote in this paper, it's quite unusual for virology paper in terms of focusing on the species. So it is because of this, what I'm going to say, it reflects mostly my personal views. Just it's not maybe common and not shared by many. So it is important to understand also. So, so virology generally, it is a very young discipline. It is just 100 years old, or 120 or something. So some people even probably alive who born when virology was founded. Maybe very few, but in the years, but it's still, so it is. And of course, as you know, so taxonomy is associated normally with very ancient something, right? So it is 18th century or something that, but taxonomy in virology was founded formally 50 years ago or something like this. So it is very new. And it is because virology was born as a kind of investigation of in the course of disease. It is always was associated with a disease and also in mind of people. And it is probably very interesting for your group because it is cross species, but also in terms of scientists, it is viewed as an extension of studies of disease. And so because of this, viruses were discovered thanks to disease as extension. But that has been changed over the last gradually in the 21st century and most profoundly during the last 10 years, and because of revolution in genomics. So the genomics now is so cheap that most viruses we know, they have no phenotype at all. So they simply just identified by sequence. And so still many virologists not easy with this. So some people, some researchers don't accept that what we know about genome sequences, it is viruses, but still it's impossible to ignore. And so the research shift in our appreciation what is viruses. And so effectively we recognize a string of the letters as a virus. And so that is very different from what we see in other parts of biological world. When we normally identify something, we see something, and so then we identify genomes. And here, all virus discoveries is going through genomes right now. So in this respect, SARS coronavirus 2, it is exception. It is not in the mind of people. I'm sure, so it's because it's traditional, but for virology, it is an exception happening when all virus is discovered by genomics, but now it is simply just emerged disease and because of this. So I could talk a lot, but I probably will probably stop. Maybe you could ask something that, and then I will continue.
Yeah, and like so we generally run this sort of democratically. You know, anyone who wants to ask a question either unmutes themselves or puts a message in the chat. But I've got a lot of questions, and part you know, towards the end of the paper you talk about the ecology of viruses, and one of the other papers we read is it just made the case that there's a massive undersampling of coronavirus populations out there. And, you know, one of my favorite parts of the paper is when you compare these different strains or types of coronaviruses to different individual people. And I love the fact that you pick Rosalind Franklin instead of James Watson. And clearly there's, so a lot of us in this room are part of a group called Feminist Science Studies. So we're keen to see how, you know, both feminist sensibilities and I think also post-colonial sensibilities influenced your pick of these different individual scientists to illuminate that, okay, we can talk about SARS, we can talk about SARS-CoV-2, we can talk about, I guess it's Middle Eastern respiratory syndrome, but they're as alike as Albert Einstein is to, you know, pick another human being on the planet. So maybe if you could talk a little bit about just the ecology of coronaviruses, you know, what we know, you know, about the origins of and the emergence of SARS-CoV-2 against the backdrop of what we know and don't know about the broader diversity of this group of organisms.
Yeah, thank you. So it's of course a broad question, and I must say that it's a very interesting question, but I would say my expertise is limited in this respect. And I have to also confirm that really our sampling is very small, and it is very biased, and biased based what we kind of look where, what type of animals or any living creatures we look and search for coronaviruses. And then, so this is what is a major limiting factor. And in other, of course, specifically in this species we are talking about, it was driven by interest to identify ancestor of SARS coronavirus. So because coronavirus field was in backyard, I mean, nobody were interested until 2003 when SARS coronavirus emerged. And so then, of course, all interest was after virus was identified to identify ancestor. And so then people looked in the bats, and so they started sampling. So that is major part of the genomic universe we know about coronaviruses. So we did not look in many other animals. So simply just people decided that bats, it is very likely a host of the virus. It could be true, but so other animals were not tested as extensively. And so, and coronavirus is, so mine, for instance, it is related to what your question, it's about ecology. So my interest in coronaviruses emerged like 30 years ago, when first genome coronavirus was sequenced in 1988. And so I was privileged to analyze first genome of coronavirus. And so we published that time interpretation of genome first was, which was quite accurate and also helped to do microbiology of coronaviruses for 10 or 15 years. So my interest was because it's giant virus for RNA viruses. And so because of this, you're interested to why and so what is behind this. So what we learned over this year is that, and this is related to taxonomy, to the species concept, that viruses of the same species easily cross species barrier of host. And so that, of course, related to your question about ecology, where is this coronavirus? So viruses of the same species can jump from one host to another host. It is simply just because they don't notice any jump. We call this jump, and it is human perspective on this. It cross barrier or something. But for viruses, it is simply just, it is environment they live in. So they easily go from one host to another host because very few changes required to make this jump effective. So, and this is what we see now, for instance, in Netherlands, when people found that many animals which live together with humans infected with this virus, SARS coronavirus. And the mink farms and the cats and dogs. So it not only in Netherlands, but also, so it shows that it is very easily can jump and so nothing. So it's, for virus, it's easy. So, and because of this, of course, when we're talking about ecology, it is very difficult question. What is a natural ecological environment for this virus? I mean, it is probably anything, almost. Yeah. But somewhere, of course, there is reservoir, what we call. But again, it is human perspective reservoir, because we approach viruses very, from human perspective, not from biological perspective, mostly from healthcare perspective, basically.
I was wondering if you could take us back to, I think you said 1988 when you published the first coronavirus genome. Where was that coronavirus discovered? What kind of host, and what brought about that moment? Why was there focus on that virus?
So in 80s, so first genome of virus was sequenced at the end of 70s, in 78. And this was Nature several papers. So simply just to show how it was difficult. And first animal RNA virus was sequenced in 1981. And this was poliovirus on the cover of Nature. And so, and of course, during 80s, people tried to sequence viruses we knew. Again, it is long before bacteria, before humans were sequenced. So this was viruses, of course. And so because coronaviruses, already people knew that it's the largest, and this is, was difficult because of this. So it took many years, and so they completed at the end of 80s. And virus which they sequenced, it was, at that time, it was the most economically important virus, coronavirus. It is it's called IBV. It is avian infectious bronchitis virus, which causes also respiratory disease in birds, and it is it's important pathogens in the for chickens for growth for chicken growth. So because of this. This virus was sequenced as this first. And so at that time, people were not interested in human coronaviruses. As I said, until 2003, nobody were interested in any medical related question about coronaviruses. People studied it from completely different perspective. Yeah, so that was first one.
And the name, so presumably at the same time, there was some kind of structural protein work to look at the receptors that look like a crown. Like who came up with the name coronavirus, and how does that story go?
So that is probably even kind of before my times. This was in 30s when virus was first virus. Actually, it was also IBV. So that is avian coronavirus was identified in England. And so when people look in electron microscopy, so they seen this crown. And so because of this, this was corona. And so for a long time, people believed that it is something distinctive. But later on, this family included other viruses which did not have corona. But now we expelled these viruses from the family, and now everything is fine. So all viruses have corona. but for like 20 years, there was a group of viruses which is called Tora viruses. which was subfamily in the family which did not have corona. But now they separate.
And maybe that's
Yes.
and maybe that's a good chance to get back to one of my opening questions is just how I guess the methods have changed over the years for designating viral species. Like the paper talks about the cross-reactivity. I would imagine early days, were they looking at different species of hosts? So now we've got genome sequencing and algorithms to help us wade through masses of data. But can you just talk over the years how it's changed? I guess microscopy, and then what was next?
Yes, it's a very important question. It's very interesting, and you're absolutely right. So technology played enormous role in the advancement of virology generally, but also in how we classify viruses. So there were several revolutions, so including like a microscope, and also ability to use in vitro system tissue culture to grow viruses. So for instance, because at the beginning only you have to infect either plants or animals, and so to see, and you look at the disease, and this is how you see viruses is there, because otherwise you cannot see anything. So that was several, as I said, several revolutions which defined how we see viruses. And so the last one is genomics. So what is important in terms of classification, when taxonomy was founded, people tried at the beginning, to have taxonomy system like it was in the biology. But virologists rejected this. And so as a result, we did not have species concept. We did not have species rank in virology until 30 years ago. It was introduced in 1992. And we still see vestiges of this kind of depreciation of species, that nobody is using species. If you look, if you talk to virologists, people don't use species. So this is what I said. I was really surprised when you approached me and you were interested in the cross-species talk. So you're first person who interested in the species and the viruses. Nobody is interested. So people look at this as a types which cause disease. So, and because of this, all classification was types, and formally it was introduced in 1992, species concept, but it was simply just as a man-made concept without any biological meaning. And because of this, people don't use it. So they say, why do you use it? There is nothing kind of useful about this. At least it's my interpretation. So that is probably complicates the whole discussion. And coronavirologists, in a way, it is driven by my group in a way. So it's because our analysis, we're trying to approach species as biological entities, and this is very different. So none other families is using this approach. And so because of this, as I said, what we kind of discussed, what I expressed opinion, it is my personal view mostly. So it's, if you would have other virologists, it would be completely different, probably discussion.
I mean, this is super interesting to me. And I was wondering if you could also elaborate a little bit more about the challenges of the species concept in a clade. And I don't even know if clade is kind of the right terminology for viruses, just because of the lateral gene transfer. You know, it seems like with the emergence of SARS-CoV-2 and so many other viruses, what you have is multiple genealogies coming together in a given form or a given type or species, however you want to characterize it. So could you just talk a little bit about the challenges of dealing with this lateral gene transfer?
Okay, yeah, so it's related questions, absolutely lateral gene transfer, and it is cited normally as a major complication to any meaningful demarcation of species. So because, I mean, you genes move, and so it's so common that it is common pool, and so it's very subjectively people say that to demarcate anything. But I don't share this opinion, at least in respect of, what is also important to mention, viruses is so diverse genetically, they're more diverse than everything else in biology. And so and also they have so high mutation rate per nucleotide, but not per genome. This is also important. Per genome, they evolve as fast as humans, not faster. But per nucleotide, and so because of this, so people look at, so you have very fast evolution, you have lateral transfer, you have recombination, so it is common pool. But what we observed in, for instance, in coronaviruses, when we analyze all genomes available over the 15 years, you have enormous constraints on evolution. So indeed, mutation rate is high, so there are lateral transfer, but lateral transfer limited to certain places in genome, very restricted. And recombination also very restricted. And so because of that, when you analyze divergence of these genomes, you could see that a continuous evolution actually splits. And so you see clusters, which you could readily identify by computer methods as a stable, and you could explain them as a action of the biological forces. Either environmental forces or biological forces, and you could find equivalent of the species of like Homo sapiens in a way. So this is why
I've got a lot more questions. I want to invite other people to jump in too, but one just directly following from that, and then please everybody else stop me from asking questions. This is very highly technical, but I've seen a lot of the papers focus on the genetic variability of the spike protein, and I would imagine that that's one of the regions that's under significant evolutionary constraint. What other parts of the genome seem to stay stable against the background of this very high mutation rate?
Yeah, so S protein, it is part of this. So S protein, it is one of the largest in coronaviruses. And it has at least two parts, like N-terminal part and C-terminal part, S1, S2, people call. And S1, it is highly variable. And S2 more conserved, and it is also correlated with how it is located in virions, so it is structurally. So, and S1 part, it is one of the least constrained part, actually. So there are other proteins mostly involved in replication of genome inside cell, which is the most strongly constrained. But S protein, in this respect, at least S1 part, it is least constrained, and it could evolve very fast. And so that is one of the, and because it interacts with external environment, it is part of the adaptability of coronaviruses. So, so in this respect, it is an example of the fast evolution.
Interesting. And that one obviously has profound consequences for what kinds of hosts the virus can interface with.
Yeah, I mean, so it is because, but it is also not restricted in this respect to coronaviruses. It's generally viruses, of course, because if they encounter a new environment, so they have to change, right? So, And so because as we know, they jump, and so it is different environment. So all proteins which interact with external environment, normally they evolve faster. So because they're constantly under selection. But proteins which act inside cell, especially in certain areas, so when they as we learned, responsible for replication. Apparently, it is happening in a highly conserved environment, in terms of environment which is very similar across the whole biological world, or at least part of the biological world. So you jump from, I don't know, from humans even to birds, and if you enter a certain compartment, you probably could find molecular environment very similar. And so you don't need to change, basically. So mutation rate. So what is important also is, so there are two types of mutations. So mutations because of the mistakes of replicative apparatus. So it's kind of internal kind of mistakes, right? And then mistakes which are fixed. So because it is not necessarily what if mistakes happen, the genome will survive. So it depends on the fitness, and so it means that constantly there is selection. A selection that could be negative or could be positive, and so if it environment conserved, it is negative selection. So all mutations which happen, they removed. And so then you could, you look and it looks like a mutation rate is very low, but it is mutation rate not low, but it simply just was removed. Because again, because also progeny is so huge. So coronavirus progeny, it is 10 power 9 and 10 power 10. So it is
from a single cell?
Yeah, so you mean number of genomes that could be generated during replication in the body. And as because of this, you have a choice what kind of to go on, because of course there are mutations there, but not necessarily every genome will survive. And so if environment constant, so everything will kind of go on like it was. But if environment changed, it could be some variant which is normally look like weird, but it would become even kind of major. And you have choice, because 10 power 10, it is enormous choice. And also generation cycle is very short. So for humans, generation is 25 years, right? Approximately, right? So this is what we're 25, 30 years. But for this virus, it's 10 hours. 10 hours, wow. 10 power 10 progeny. And so then you have choices. So that is a major difference.
Thank you. This has been really illuminating. And I'm gonna just leave an awkward pause here and let other people jump in. I know there must be other people besides me with questions. I'm kind of zooming. Astrid, I think you have your hand up.
Yeah, hi, thank you very much for these explanations. I wanted to come back to a comment you made earlier about the change of perspective, to look at the virus as a disease-causing agent, or look at it, or at least look at the genome as a biological significant entity. Can you elaborate a little bit more? Not really what caused this change in perspective, but how does it manifest itself? Why? How do you look differently at the virus as a disease-causing agent? What are the significant features that you want to know about the virus as it compared to the biological entity, or whatever the view was that was pronounced before this shift happened that you mentioned? Does it make sense?
Yeah, I think it is a very important question. And I must say that it is happening. It did not happen. It happened for some people, but probably not for 90% of people. Yeah, and definitely not happened in society. So, and it happened just because of technology advancement, not because kind of scientists wanted to look at this. So we presented with this opportunity. So we now have genome sequences. As I said, we discover viruses because of genome sequences, and we have to explain these genome sequences if we recognize this as viruses. And effectively, what we kind of have now in virology, we have research on biodiversity of viruses, because we have no idea about phenotype of these viruses. So and you have, of course, genomic diversity, but behind this genomic diversity is biological diversity. So in this respect, it is as anything else in biology. But that is not how virology is seen from outside. So people ask, so what this virus causes in terms of disease? But we don't know. I mean, it may not cause disease at all. Yeah, so it's also important to realize that it is really human perception of viruses about disease causing. So because even with SARS coronavirus 2, it is very benign virus from biological perspective, because we don't know really a mortality rate, fatality rate yet. But it could be like from 0.1%, but probably something between 0.1 and 1%. From population point of view, it is nothing. So if it happened not to humans, it happened somewhere in the nature, we would not have noticed at all. Because in nature, species are kind of killed at the rate 20% of population often. So it is jungles. It is constant fight for survival. So 0.1%, it is nothing. But of humans, of course, it is absolutely different, because we care about every life. And so because of this, we look at this virus as a pathogen, but it is not pathogen. It is simply just our human perception. And also a disease, it is a result of interaction of virus and as a body and human body. And that because disease is caused in 0.1%, it is really difficult to study, because it means in 90% nothing happens, nothing wrong. So it means that there is in specific persons, there are certain conditions which, or maybe also conditions which are result of dose of virus or something other environment or something else, which cause disease or even mortality. So as I said, it also creates huge problem to study this. People don't acknowledge normally, you don't hear this, but you have to reproduce in the lab something that it happens in 0.5%, but you have to scale up to 100% to study this. But then you end up with some system, artificial system, which doesn't reproduce what is in nature. So, and the question is, what you learn from this system, how it is applicable to what is happening in nature? It is question mark.
So Francisco has some very smart questions in the chat, ranging from one that I had too, where is the type specimen, if we can think about it like that, of SARS-CoV-2 physically located, you know, in other branches of the phylogenetic tree. You know, beetles are, you know, encased in gold and stored in collections. Is there something similar for viruses? He's also got some questions about the species concept, about bad ecology. So Francisco, do you want to ask those yourself?
Hello, everyone. Thank you for this chance. Nice to meet you, Dr. Gorbalenya. It has been very interesting so far. So even what you have just said is probably enough to characterize what I had in mind. The first question, as you were saying, is important for any of us who have described species. We know that we have to prepare a type specimen and put it in my case, I work with plants, in herbaria. So in the case of virology, I was interested in knowing how do you deal with that, with type specimens and other nomenclatural issues?
Yeah, thank you. It's an important question, and it's an unresolved question, if I correctly understand. And that is also one of the main causes why some virologists is still not happy with recognizing that sequences could be recognized as viruses.
Right.
So it happened. But again, it happened by International Committee of Taxonomy of Viruses. So this is why I'm saying it happened, because ICTV, because it has to classify viruses. And so it was confronted with the questions that we have mostly viruses, new viruses, genomic sequences. And so you have to recognize either you accept them for classification or you don't accept. And so, and effectively, it means that you have to recognize that genome sequence is virus. Although technically speaking, ICTV is not empowered to decide for virologists that sequences is viruses. But because it's recognized that it could be classified, so it recognizes as viruses. But of course, people, only few people involved in taxonomy issues. And so some people who are still doing experiments, they say, no, I mean, I would accept viruses if I can put them in fridge. And so I would see something that really. Right, so this is a question, as I said, different people decide differently. And so one of the problems with genome sequences, in addition that it is not something that you could fridge, and it also we don't know what is disease. Often we don't know even host of this genome sequence. So even more difficult. We don't know what species this virus infect. So some, of course, most of these viruses were isolated, genome sequences were isolated from certain hosts. But there are some compounding issues there that it could be contamination, for instance. You don't have evidence that this virus infected a host from which you took this specimen. And so because of this, we don't know even if you would like to study this virus, you don't know even what it infects. Even often you don't know whether it's a plant virus or a mammalian virus or insect virus. And so it is one of the questions so for computational biology. So people try to develop tools and also some identify features which could be recognized in genome sequences. It could say that to associate with host. And this would be a great step forward. Yeah, so
you have already started to say something about your own stance regarding species concepts. Could you say a bit more about it, considering the variety of species concepts that botanists or zoologists or other biologists actually entertain to describe species themselves, and also philosophers of biology have been nagging about this for many years also.
Right, yeah. So technically how we do this, so we use all genome sequences, as I said, when we started doing this, and it happened after outbreak of SARS coronavirus 17 years ago. I was already got interested in the classification 1 year before, and people trusted me because for as I said, 13 years before, I did computational work with genomes for functional annotation of coronaviruses. And what we proposed, we were quite successful with this analysis. And so people trusted because they've seen that computational analysis could be verified in experiments. And so because of this, next question was, okay, so could we use computational analysis to make classification of viruses? And so we started with full genomes of all coronaviruses, and we didn't have any preconceptions. So what we asked whether we could find, if you look in the, so evolution that happened, so you would expect that accumulation of mutations, so divergence. So whether you could see some discontinuity in the spectrum of distances, evolutionary, if we could do this. And indeed, when we develop methods and also criteria and statistical apparatus, how to do this, to also to address question of biased sampling, because it is enormously biased sampling, and so one of the questions. And also to account for multiple substitutions, because it is a lot of substitutions, and so just to give you idea, because it's nothing like this you will see as in other areas of biology. So one position on average could be replaced by 5-10 times. So it is impossible, because of this, impossible to say anything about percentages, because in percentages scale only 100%, and here you're talking about 500% or 600%. So it is very difficult. To make the story short, so what we observed discontinuity, and so we were able, if we analyzed most conserved proteins for which we could align all these genomes, we observed clusters of viruses, and these clusters had biological sense. So we completely did this without regard any biology, without anything knowledge about anything experimental. But when we presented this to coronavirologists, and even broadly more to other, so coronavirus is related to several other groups of viruses, which is called nidoviruses. And so we have meetings every 3 years. So people found that it's quite sensible. And so we continue to do this. So sampling increased, sampling since when we did first time increased probably 20 times. And still we see species, and so we established by this in this way species and genus, and so family, and so all this kind of taxa on different ranks. We've seen stability of clusters we identified genomic, and so this is how people use. And on the lowest level, on the species level, clusters we identified viruses inside cluster could recombine, could exchange. And so this is, you would say, it is equivalent of the sex in the humans. So that you could, progeny could be as a result of mixture of two parents. So the same is true for coronaviruses inside species identified by this method. But between viruses belonging to different species, we don't observe recombination in the most conserved proteins. They could recombine in other areas, but it is would be equivalent to horizontal transfer or something. Because also I have to mention that horizontal transfer even happens in humans, but still we recognize humans. We could recognize humans, despite that they also recipient of horizontal transfer. So it is not a kind of fundamental problem. So question only extent of this horizontal transfer. Okay.
I have all kinds of questions from that, but I also, I mean, for me, it's fascinating that, you know, I think it's 8% of the human genome is viral origin, at least according to some studies. But I also wanted to elevate Francisco's question about bats in particular. I know that a number of bat ecologists are saying, because of this massive undersampling, we can't point to the bat as the single point of origin. And I think there's animal welfare consequences, because you know, some people are just trying to kill bats right now, many of which are endangered animals. So I was wondering if you had any thoughts on just the sampling of bats, and how just looking back at what happened after SARS 17 years ago, was that bat sampling effort, you think, too narrowly focused, or, you know, should other animal taxa have been included, or what do you think?
Yeah, it is an extremely important question moving forward. It's just because what you're saying, it also after this pandemic, also there would be certain actions. Also, people try to make life safer, and so that is probably the major problem. not with bats. It is how we approach our life. But it is, of course, a bit different perspective, and also it's more difficult to change than to kill bats. So people look in bats because there were indications, because I said, like with this outbreak, initially 17 years ago, people linked the whole outbreak to the market, animal market. And so when they looked, and so they identified viruses which were closely related to viruses which were isolated from humans. And so, and bats, there are several considerations why people look in the bats, because it is the most numerous animal mammals, most numerous species mammals. Yeah, so it's like 300 different mammals. Also huge populations living in proximity to each other, so that it is easy to transmit viruses. So it's, there are considerations, and also in China there are many, although bats, of course, everywhere, United States and Europe, so everywhere you could find. But proximity, of course, living proximity, it also this wild animal markets in the China, so they might. So people tried, and so this particular one researcher in Wuhan Institute, she's of course in the news for the bad reasons. She, her name, and so she looked specifically at many kind of caves and the bats, and so eventually she was able to identify virus which was very, very closely related to the virus which we isolated, people isolated from humans. But there are still differences, and because this virus was isolated from bats, it is not ancestor. So we don't have ancestor because it was, but it means that you could, based on the distance in the nucleotides between these two viruses isolated in the bats and the humans, if you apply certain methods and assumptions, you could say, okay, so they diverged like, I don't remember exactly, but could be like 20 years ago. So or something, it's very recently. So still, of course, you cannot ever exclude, especially because virus is so easily cross species barriers. So there is another species of host where the same virus exists. But simplest hypothesis would be say, okay, so that is the descendant of the common ancestor, and that you could assume that this virus circulates in this particular species of bats, although you can't exclude that there is another species of bats which could be kind of also shared with this virus. So that is what happened 17 years ago. So there is quite strong evidence, I would say. But we will never know for sure. And it would be impossible probably to reconstruct the whole exactly event when this jump happened. But in this case, difference is bigger, but bats, the same species is there. So people naturally suspect the same type of host as a possible reservoir, what they say, for this virus, for natural reservoir. But again, probably there will be always some level of uncertainty about reconstruction. Because for complete reconstruction, you have to isolate ancestor, exactly this virus, but it is very unlikely that we will be able to do this.
So Anne's had her hand up for a while, and Rachel just had a question in the chat. And I should say that if I'm going on my phone today, so I can't see everyone. So if other people have raised their hand, please message me in the chat. But Anne, go ahead. Oh, I think you're muted, Anne.
Yeah. You have to switch on, yeah.
Thank you, Professor Gorbalenya. Thank you for this very informative paper. So as I said earlier, I'm an anthropologist by training, but I just recently wrote a paper with a historian called Multispecies Entanglements in the Virusphere, Rethinking the Anthropocene in Light of the 2019 Coronavirus Pandemic. And so we took the case study of SARS-CoV-1 because, as you just said earlier, it is not, we know so little about SARS-CoV-2 yet, exactly what the mortality rate is or where it exactly, where was the origin, was it a wet market in Wuhan or was it somewhere else or was it in a laboratory? So we took as the case study SARS-CoV-1. And from our understanding is that these pandemics will become in the future more frequent because there is anthropogenic climate change and overpopulation. So in a sense, there is a homogenization of the virusphere and the friction zones between the virusphere and the anthroposphere is increasing, making it more prone to have pandemics to happen in the future, and not only every 10 to 40 years, but maybe more frequently. So my question is, what are the biggest challenges in taxonomy of for viruses these days? Because I would assume that changes are being made in response to new information on known and novel viruses, and the rate of, since there is more and more of these viruses and so many haven't been classified, are there analytical methods based on genome sequencing and maybe automated classification in artificial intelligence that can help and assist with classifying these viruses? Yes, and maybe AI can also help when species needs to be sorted and when classification and nomenclature are being blurred. So my question basically is, in what way can AI help in the taxonomy of viruses? Thank you.
Yeah, thank you. And so you actually raised many questions, and it's very important questions at the beginning. It's especially when you said that pandemics could be much more frequent than because of, as I said, it's we share this viruses, everything, and now. And because all this pandemics, but it depends whether we will learn from this. If we learn and we change our behavior, pandemic may not be as frequent. So, but it is of course much more difficult than to kill all bats. So it would be much easier to kill everything around. But it will be a reactive approach, and so it is a test for humanity to go forward, but they could keep civilization and advancement and interaction with nature intact or improved. And I must say that because we have already 4 human coronavirus circulating, so they entered a human population before, and we don't know how it happened. And pandemic could have actually were even more severe than what we observe now. So we simply just don't know. And because they now benign viruses, I would say that it also shows that what we kind of face now, it is simply just one-time event. So after all people vaccinated or infected, our interactions with this virus particular will be much more kind of peaceful. I would say that what we experience now. So it's also a possibility. So answering your question about taxonomy, so there are several issues here. So first issue, it is taxonomy classify what is available, genomic sequences. And our sampling, although it is very biased, it's extremely small. So if you look in the biological world, people easily say that we already know, and they classified 100,000 species simply just because they were described. I'm talking about vertebrate, invertebrate, plants, and others, several life forms. But with viruses, we have genome sequences, and we have only 7,000 species. So some, of course, genome sequences have not been classified. But if we assume that everything classified, we would get probably like 10,000 or 15,000, but it's almost nothing compared to what we expect, because we expect like 10 power 10 or 10 power 11. So it means that problem now is not with taxonomy, but it's simply just with genomic sampling. It's extremely small still, because we did not test. But taxonomy, of course, will play important role, because it organizes data, because otherwise it's genomic sequences, and it is created, it structures them, and so then people could approach because it's classified. So then it's entry point for different for different researchers and also for public, right? So because then it's names, everything. So and here we have a major problem, because taxonomy of viruses is being conducted completely different ways than taxonomy everywhere else. So for virology, we have a committee which oversees taxonomy development. We don't have anything like this in plant, in animals, or anywhere. So they are researchers doing taxonomy. They discover something and they classify. So with viruses, we don't have. We have a hierarchical system. There is a body that's empowered to endorse. And so, and that is for the reason was made, because viruses was few, and they was considered so important, and so then they created a very complex process. Originally, it was 3-5 years to recognize a new species. Nowadays, it's faster, but it's still 1 year. And again, it is people who discover, so they submit proposal, and so then it's executive committee considers them, and so then ICTV, so there is several hundred people who again approve this or disapprove this. So, and all these people are not paid. So it's absolutely voluntary system. And so, and more now sequences coming, and analysis becoming much more complex, computationally intense. So it is a major problem. So there is no curriculum in any university in the world on virus taxonomy. So there is no one grant on virus taxonomy in the world. And then, and still, how kind of you have to organize it. And so everybody, I mean, not everybody, but at least some bodies look for taxonomy because it organizes data. So, so we have a gene bank where genomic sequences, and they organized according to taxonomy developed by CTV. So it is major driving force. But gene bank is funded by American government through National Library of Medicine. But ICTV, as I said, there is no funding. So it absolutely people who devote all their time, and I don't know how long it will survive, this system. Okay.
It seems like a problem of taxonomy in every branch that I've spoken to herpetologists, plant taxonomists. It seems like taxonomy is not sexy to some funders for some reason. I wanted to bring in Rachel's question, and it also related to something that I was thinking about as you were talking about these 4 major pathogens. At least from talking to physicians, it seems like coronaviruses are circulating in the population all the time. So there's kind of this background diversity of human coronaviruses. Rachel, do you want to ask your question? I mean, it seems related. You're asking about currently about the expanded diagnostic testing in the context of understanding the ecology of these viruses. And for me, I would think that the ecology would include these more common cold viruses. Do you want to jump on, Rachel?
Yeah, just thank you so much. Just thinking about this, what you talked about earlier in the article about the disease, the sheer just disease spectrum, clinical manifestations, species manifestations. I was struck by the very last line of the article which was a political and economic one. To advance such studies, innovative fundraising approaches may be required. And I was certainly struck by this as somebody who's teaching biotechnology and thinking more about the tools that you talk about, complementing the existing tools. And you also discussed particularly thinking alongside the already impressive advancements in personalized medicine in recent years. So I was kind of struck by these things, and was just wondering, what does this endeavor of expanded diagnostic testing in light of the ecology of these viruses? Dream world wise, and again, I'm coming at this as a social scientist, that what does that require? What does that look like? What's your ideal scenario? If you could just talk a little bit about that, the endeavor of that expanded diagnostic testing and the innovative fundraising approaches you could imagine or would like to see.
Yeah. Thank you so much. First time I hear somebody read this part of the article, which we added at the revision, Nature, at the end. Right, yeah, so it is, I believe it is an important question, and I don't have answer. So this is what I simply put this because I saw that it's important to think about in this direction. And so problem is that it's as always in our life, it is money. Unfortunately, money, and we already discussed with taxonomy, but money is here. And so it will require, it will increase cost. And how much, it depends what we can afford. And so as society. But a problem is viruses, as probably, especially in this group, you recognize. They don't recognize borders, not only borders between species, but also between countries. So that is a problem. So something happened in China and major economical actually consequence in the United States. And we see kind of in the news discussions about this. But and then you have to, if for instance, could you imagine, for instance, if it did not happen, we would not have this type of discussion generally. So because people would not entertain simply just a theoretical possibility of this. Now we kind of facing this. Of course, it's at least it's the moment we could discuss, and I hope that at least people who is in power, who decide about budgets and long-term budgets. So the question, it's not about 1 year, 3 years. You have to allocate for the life. And so then it's, of course, if you allocate money for this, so you have to take from somebody. And of course, politicians have problems because they don't have enough money already to satisfy all requests from society, other kind, not about viruses. So this is a really kind of question, how much kind of you have to allocate to this. And so for instance, to give you example, which could have done already, but it never been done. So for instance, all viruses which cause disease in humans, they never been tested in in healthy population. So for instance, often people compare consequences of this pandemic with pandemic caused by influenza. But we cannot compare because we don't have numbers about the rate of infection in healthy population for influenza. We don't have mortality rate for influenza for the whole population. So everything is done based on the people who admitted to hospital. So at first time in the history of virology, people tested for virus which don't have any signs of disease. And so that is give you kind of idea about, for instance, if you kind of introduce testing. So it is this type of testing, of course, you would need. And this is very unusual, and also socially, its challenge would be for many people, right? So how, I mean, I'm not sick, so why I have to be tested? So for instance, and because of this, we have no idea about like how coronaviruses, for instance, circulate in human populations. Because people only if you sick, you are tested. But of course, evidently, virus circulating not only in sick, so everywhere. Again, so it is related in the equations because what we raised there and we gave examples. So for instance, if in China or in other places there are RT-PCR tests directed not against SARS coronavirus, but to the species of this virus, they would have recognized the SARS coronavirus 2 immediately. So that's what we pointed. So you have to make species, not virus lineages like it's now, as a target for diagnostic. But it would be much more expensive. And chance of the pandemic is so rare. So you have to invest billions to prevent event like this. So when you calculate cost, you have to calculate cost of this type of event. You prevent trillions of dollars and distributed globally, basically. But which government will take on this? I don't know.
So that relates directly to Melanie's question, and I should say normally we go an hour, but I'm finding this discussion to be really fascinating. And as long as you have time to keep listening to our questions, Alexander, I'll keep it going. So is it okay if we take a few more questions?
Yeah, right. Yeah, no, yeah, I appreciate it. Yeah.
Wonderful. Yeah. So Melanie, who's joining us from South Africa.
Thank you so much. Yeah, I think you partly addressed the question already. I was also struck when reading the paper that you mentioned the need for innovative approaches to funding. And yeah, I mean, how important is it to do the research you're doing on a global scale and to collect the data you're working with on a global scale? And you mentioned the lack of funding and that it is concentrated in the US. But I mean, how important is it to cooperate with researchers, institutions in the global South, like Africa, South America, etc.?
Right, right, yeah. No, it's. I mean, if you ask me, of course, I believe it's important. But we're dealing with all these realities of this world, right? So which is very fractured and so unevenly developed. So and simply just virus reveals all this type of problems. So because when you suddenly when we don't have borders, we see what are our problems actually. We look as a mankind, right? So because normally we restricted by national borders or even less so. So I mean, and we have WHO, which is on the mercy of governments, very difficult position, but also with very restricted mission. So we don't have so anything to create above governments, it is very difficult to sell to population in the countries. So we don't have mankind. We have mankind globally, of course, living. So, but we still we have lineages, restricted geography, and by political borders.
So Stefan asks a question, do we know anything about chronic infections by coronaviruses? And I guess that also goes back to my question about sort of the natural history of the human cold. Are we constantly kind of living with kinds of coronaviruses that are similar to SARS or SARS-CoV-2? Or do the kinds of chronic infections that are constantly circulating in the population, does that represent a divergent lineage? I'm not sure I completely understand the question. So if you're talking about species to which SARS coronavirus 2 and SARS coronavirus belong,
So we don't have any other lineage which is in the human population. So in this respect, this virus species second time entered human population. First time unsuccessfully it entered 17 years ago, and second time it's now entering population, right? So it's the same species, and we don't name it species.
Then I might have bad. Do I have bad information then? My sister, who's a physician, told me that we're constantly being infected with coronaviruses, like the common cold. Is that not the case?
No, it is a case, and so we have several problems here. So it is because of this. So when you kind of watch TV or read newspapers or something like mass media, so people either talk COVID-19 or coronavirus. And so both of them, it is misrepresentation what actually happened. Because COVID-19, it is disease, it is happening in a fraction of people, of course, it is not virus. And coronavirus, it is something very general, because there are many different coronaviruses. So just to give you example, so nobody talk, when we talk about influenza, nobody talk orthomyxovirus. You probably never heard about. Or Ebola virus, nobody talk about filovirus. And so that would be equivalent to coronavirus. So when people say coronavirus, they already misinform kind of discussion, because as you said, that we have 4 species of coronaviruses which circulating constantly in human. And probably in many cases, we have a double or triple infection by several coronaviruses, several different type of coronaviruses, or different species even coronaviruses. So not naming, I understand that it is convenient to use coronavirus because SARS coronavirus 2, it is long. But if you name a species, it even longer. It is very unfortunate because it's severe acute respiratory related virus. So that is how its species is called, and nobody is pronounced because it is so difficult. And for others belong to different other species. And some of those species actually includes viruses which infect different animals. So like this species include viruses which infect bats. So in other cases, for instance, there is human coronavirus OC43, and there is bovine coronavirus, and they belong to the same species. So what does it mean? That human coronavirus OC43 can infect cows, and cow virus at certain conditions could infect humans, and they could recombine. And very rarely, probably because it's simply, but they belong to the same species. They're so similar. So it's important to realize. And of course, this virus causes a similar disease in cows, respiratory tract. So it's yeah.
Super interesting. Stefan, did I get to your question, or do you want to ask it in a slightly different way?
Yeah, no, it is part of the question. Just another part was just often when we talk about common cold viruses and these other kind of coronaviruses, it's always this picture that we just catch it and then we completely clear it, and we don't, the single individual doesn't carry it for long. But with other viruses, we keep them in our system, like really on a person by person basis, much longer. And I was just, because with this kind of little knowledge about how asymptomatic people, what do they carry, how does the virus circulate in the larger ecosystem? I was just curious to know whether for any of these kind of coronaviruses, very broadly speaking, not SARS-CoV-2 or anything, but just what do we know about these types of viruses, how they interact with single humans? That was kind of, are they chronic? Do I kind of have asymptomatically for years kind of carrying? Do they find a balance with our immune system and they kind of coexist or not? Or that was kind of part of the question.
Right. Yeah, it's a very important question. And because I'm not, I was trained not as a medical doctor, as a biologist by training. So I could answer only in the kind of in my my expertise. So when I answered to question to Rachel, so I already mentioned that we don't have any idea about circulation of these viruses in a healthy population. And so that is exactly would be important to answer your question. So people understand, all our knowledge normally coming from diagnostic departments of the hospitals. And diagnostic departments given samples of the patients coming to hospital when they have some manifestation of disease. So healthy population were not tested. So we don't have any idea where this virus is circulating, which is called common cold. And because they circulate in human population, it apparently, as I said, what we observe now with SARS coronavirus 2, it is probably typical for other coronaviruses in only in few percentages. We don't know in what percentages because we don't have data. We have a manifestation like a common cold, but many others probably even don't notice anything. And so because of this, as I said, if you look objectively, viruses not pathogens. Most of viruses not pathogens. So they simply just part of our environment. So they live with us. And so, and there are many examples when they are useful. We simply just don't know and we don't understand this. But they part of this. And of course, still a question you asked about how long you could carry virus. It is important if you can. Okay. Some viruses, not this type of viruses, not coronaviruses, probably not rhinoviruses, which is also called common cold. So common cold could be caused by different species which belong to different families, which are different as, for instance, humans and yeast. So viruses is so different, so that it's enormous, and they will still cause common cold. But mostly of them are RNA viruses. But there are DNA viruses which infect everybody, and they, once they infected, they stay with you the whole life. So like herpes viruses, polyoma viruses, many. So once you infected, to the very end, they're with you. And so they called opportunistic infection. And so in certain cases, when organism is weak or something, they could cause complications, but normally they nothing, so there is no effect. But it's unlikely with coronaviruses. So it is called, it's called acute infection. Normally it's released. And then it go to another person.
So Francisco in the chat has introduced a really important concept that is, and it started to basically prompt anthropologists to rethink what it means to be human. This biological idea of the holobiont. And Francisco, do you want to ask your question? It's related to you know, how some of these things that you were just talking about, Alexander, might prompt us to really think the human microbiome and the human virome in terms of that holobiont question.
Well, as you say correctly, even I was just trying to connect some of the philosophy of biology topics that have been recently discussed a lot, this issue of the holobiont, as you mentioned. I was trying to connect that to what Dr. Gorbalenya was just describing in terms of the inescapable multi-species nature of each human body, right?
Right.
Do you have any thoughts? And maybe this is a very big picture question, and maybe a good one to end. This has been a delightful discussion.
Right. Yeah. So of course you already mentioned this, and also you formulated. It's an exciting development which happened, probably started in 21st century when recognized microbiome. First it's microbes and bacteria normally, but now viruses included. Viruses which regulate bacteria, phages mostly. but we're probably also other viruses. And we have no idea, or at least we have very kind of scratched the surface only about our interactions. So humans as ecosystems, right? So it's we symbiotes.
Yes.
Of course, we have brains, and so we represent, and we talking about we believe that we know who we are. We look in the mirror, and this is what we are. But of course, we see only part of the picture, and we don't know what other parts which we don't see, how much it affects our self, kind of what we are basically, how to make so. And also it connects actually to external world, because they kind of move easily, because we as a solid piece, and they much easier to enter our body and to leave our body. So in this respect, they connect us to literally to ecosystem at large. Yeah. So in this respect, I mean, you position it much better than I to think about and to realize this. And it's fascinating that you started this club and thinking, and I understood that already more than 10 years ago, thinking about like multi-species concept. And yeah, I really admire what you're doing.
Well, we admire what you're doing too, and it's been a really enlightening chat. So I hope to keep the dialogue open, and if it's okay with you, we'll put your email list to get our weekly announcements. So if you want to come back, you'd be a very welcome guest in future meetings.
Yeah, thank you. Yeah, thank you. Yeah, so it's yeah, that's thank you. Yeah, so I would appreciate it.
Thank you.
Okay, wonderful to chat, and see you all next week.
Yeah, yeah, thank you.
Thank you.
Thank you.
Thank you for all your questions.
Bye. Thank you.
Yeah, yeah, good luck.
Thank
with
you very much.
Thank you.
Bye.
Thank you.
Bye. Bye.
Thank you.